And what this question does is it integrates a lot of information that I've been assessing over time that there's still some misconceptions in the classroom about. Almost all of you, vast majority of you, got question 1 correct. These are metaphase chromosomes. And when I say write a one-sentence answer, I really mean it. When you see the exam on Monday, the amount of space I leave underneath a question should give you some idea about how much information I'm looking for. So you don't have to write paragraphs about something if I give you that much space. So this is metaphase. Why? Because all the chromosomes are condensed. This is the only time in the cell cycle we'll see chromosomes as individual objects. What do the chromosomes look like when they're not in metaphase, if you could describe it in words? When it's any other point during the cell cycle, growth 1, growth 2, synthesis.
Like a bundle of DNA skulls?
Like a bundle, like
A bunch of DNA strands?
DNA strands. In my vernacular, the DNA might look kind of like a cloud almost. It's this sort of decondensed thing. You might have one chromosome 1, for example, let's say. Looks like it's blue. There might be another region in the nucleus where chromosome 2 is looking like this. Chromosome 3, chromosome 4, chromosome 5, both copies. It's just fluffy cotton balls of DNA. Now, where things fell apart a little bit was question 2. Which is always a huge issue for genetics. How many copies of chromosome 5 are visible in this karyogram? So what sort of answers do you think I got? I got people saying 2. What other answers? Fortunately, nobody said, I don't think anyone said 1. If they did, I didn't read that one. But that would be another. So it's either 1, or it's 2, or it's none. That's right. So there are there are copies of chromosome 5. Thank you very much. Wrong answer. So this is one where there is at least possible wrong answers. It's 4. And this answer, 2, has come up. I've asked this question in various ways 3 or 4 times now during the semester already. So this is really important to go over again, which is why I'm taking up your question asking time to do this. I suspected this was going to be the case, which is why I made this assignment to talk about today in the review session. And the rest of these questions are related to this point, so I'm actually going to come back to question 2 at the end.
Question.
Please.
Could it possibly be 4 copies if we know it's a human karyogram?
And what do you know about humans?
23 pairs.
23 pairs.
Yeah.
Right. Diploid. This is where the source of confusion comes, and that's why we're here right now. So that's an excellent question. It's right on point. How could we have 4 copies of a chromosome, which I said was the right answer? That's 4 double helices of chromosome 5 we're looking at. If we're diploid, how do we have 4 copies of something?
The cell just replicated the DNA and hasn't divided yet.
Right. So the solution is, what stage of the cell cycle are these chromosomes? We all agreed it was metaphase. What's our ploidy at metaphase? This is after DNA synthesis. After synthesis, we're tetraploid. The copies of the chromosomes that we got from, so we've got a copy of chromosome 1 from mom. It has replicated by the time we get to metaphase of mitosis. This is right before cell division. So we've got 2 double helices, both of which came from mom. That thing is that object. 2 chromosomes lying side by side. You can't even see that there are 2 there in this picture. And if we looked at the copies of chromosome 1 from dad, after synthesis, so we have 2 copies, that thing is that, 2 chromosomes lying side by side. So in this picture, 4 copies, 4 double helices of every one of the 23 chromosomes. Questions? Anything? On this, certainly, now we're open to questions. Review time. What do you want to know more about? What would you prefer to know less about? What concerns do you have? How can I help?
Dr. Ross,
Yes.
you said during the week that we would have more time to do the fish experiments.
Okay.
Uh,
Have you looked at the study guide? Okay, so there's one fish question that's in the study guide, right? So important, I'm glad you asked. Reminds me to mention, if you hadn't seen the announcement, study guide is on Blackboard. About 3 of you participated, which is actually kind of underrepresented. Usually, in most terms, a lot of students send potential questions in the study guide. This time was pretty low turnout. Please be engaged in this process, because it will help you study. So I threw in a lot of questions I would. So one of them is a fish interpretation question. And I've also posted the key to the study guide. So the study guide itself is a PDF, it's just questions. It's like a mock exam. I recorded a key that's a video now to that study guide, and it's on YouTube. So if you want to know the answers, you have to go somewhere else to get the answers. It's not as easy as downloading one PDF and just being able to scroll to the next page and seeing the answers. So study guide does exist. It does have one fluorescence in situ hybridization related question. It has a few questions about if the order of the chromosome fragments in fish is A, B, C, D, and you wind up with a chromosome that's D, E, F, then what process do you go through, inversions, translocations, deletions to get there? There are a few of those questions. There are a few other types of questions on the study guide as well. Should we do a fish question now? I could probably make something up on the fly.
Why don't you look at like all of it, or like the whole? On the study guide, you ask a question and you give like a DNA sequence and you ask like the species and stuff. Would that be a test question where we would just put it on blast and like learn that information, or is this just for our practice?
So, okay. So the study guide contains a question where I give a DNA sequence and I ask you, tell me please, what species is this from, what chromosome is it on, what gene is it part of? Which is very similar to the in-class activity we did last class, interpreting blast output. That's very much something that I might ask on an exam. The exact same type of question, I could do it.
Using outside resources.
So, using outside resources. Does anybody have any questions about the format of the exam? It's kind of a loaded question. How many people would like to know what the exam is going to be like? Yeah. Why didn't you ask that first then? Okay. So I'm going to what I'm about to tell you does not have to stay in this classroom. I'm actually going to post, I made myself a cheat sheet. I'm going to post all of this on Blackboard. So I'm going to give you some highlights. Read this when I post it electronically later. It tells you a lot of detail about what the exam is going to be like. But yes, I told you on the first or second day of class, actually, I think, that I was hoping to do group exams, open everything resource exams. We're going to incorporate both of those in the exam on Monday. Don't freak out. Everything's going to be okay. I promise. Seriously. Unless the internet totally goes out in the building, in which case I will improvise. It's not your fault if the internet goes out. So no one ever gets punished for technical issues. Yes, sir? Okay, so the one question I'm not prepared to answer right now is how many questions? It will probably not be, so, and as I said on the second day of class, my testing philosophy is I want to give a number of questions at multiple levels of critical thinking. So there will be some questions, and remember the breakdown of points to get letter grades in this class. 0 to 20% of total grades gets you an F, 20 to 40 is a D, 40 to 60 is a C, 60 to 80 is a B, 80 to 100 is an A. So you can expect that 20 to 40% of the points maybe test factual recall. They're easy questions. They might be multiple choice, true, false, fill in the blank. This is an indirect answer to the question of how many questions will there be. There will be a number of questions that are probably pretty fast to answer. Maybe you could just look them up in the textbook because you've got your tablet with you for the exam. You've got internet access. You've got Wikipedia and everything else that's on the internet. But you might spend all of your time looking up information, answering those easy questions that only make up 20 or 40% of the grade. So maybe you get yourself to a C just by answering factual information. The rest of the exam, probably fewer numbers of questions, maybe 1 or 2 or 3, are where I try to figure out, can you apply information in new situations? Can you interpret data? Can you make predictions? If I explain a scenario and I say, what do you think would happen? So it's higher level critical thinking skills that'll make up another section of the exam. So my advice is, well, you can read this later. I'm not going to waste your time telling you. You can read this. I'm posting advice on how to study for the exam on Blackboard. Please ask if you want, but ma'am.
How many per group?
How many per group? It'll probably be 3 or 4. I'll let you choose your groups. If people aren't in a group, so here's the way it's going to work. There will be 15 minutes. Everybody does an individual exam, electronically on your tablet. So bring your tablet fully charged. No tablet, no exam. So 15 minutes, 80% of the total exam grade. So 80%, you get up to a B grade amount of points just from your own work. 80% of the total value of the whole exam is individual work. You, with your tablet, doing your own work, sitting quietly by yourself like a normal exam. Okay, that'll be 15 minutes, the first 15 minutes of the exam. At the end, I will turn off submission. So you have to have submitted by the 15th minute. And there will be a timer up here so that everybody will know exactly when you have to have submitted by. Don't be late.
Wait, so is that part gonna be like the kind of classroom?
It could be Socrative, it could be Qualtrics, one of the web-based surveys that you've taken. It could be something, it could be PDF annotation. Anything we've practiced doing in class could be a Google assignment, annotate a PDF, upload it to Google Classroom. Anything we've done in class is a fair approach. So that's the first 15 minutes of the exam. So you don't have very much time to answer who knows how many questions. 80% of the grade. 5 minutes then to form groups. So you don't have to come with a group. If you want to form groups of 3 or 4 in advance and come ready with a group, that's fine. Those 5 minutes will let us move people around to sit together, and for me to form people who don't currently have groups into groups of 3 or 4. Okay, so 5 minutes in the middle of the exam will be us moving around. Then there will be 25 minutes of group work. I'm not going to tell you what that's going to be like, but I can tell you that I have methods of assessing the quality of groups and taking that into account. So this is right, this is what everybody worries about, it's what I worried about when I was a student when I had to do group work. What about that one person that just tags along and gets the grade of the group even though they didn't do anything? That will be accounted for. So you do, there is, and that's why the group grade is only 20% of the total exam points. Because some of you maybe aren't comfortable working in groups. You're shy or you're an introvert like me. You don't like to talk, which is An introvert does not mean they don't like to talk. It means we like to think things through before we say them. And in groups, that doesn't work for introverts. So maybe you're an introvert. You still need to participate in helping the group as a whole discuss whatever the question is on the group exam, question or questions. At the very last 5 minutes of the class, so that's the group exam, format to be discovered by you in the exam. The last 5 minutes will be you giving me feedback like we normally do for an exit exercise. This is the point where I'm going to ask you to do things like rate the contributions of the different group members. So it's going to be peer assessment. Who rank number 1, 2, 3, 4, who contributed the most, second most, third most, fourth most. I will not use this to penalize. If everybody agrees that one person did the least amount of work, that person will not be punished. There won't be fewer points for that person. I might elevate some of the others. There will be benefits to those who get ranked highly, especially across the term. Like I might ask you to come back and help me teach genetics in the future. If you show that you're good at working in small groups, and by the way, I'm wasting your time. Last thing I want to say about this, I'll post this later. I'm doing this because a lot of you are going to ask me for letters of recommendation before you graduate, and I want to be able to say, in part, this is partly the reason that I do group exams, I want to be able to say, I have observed a student working in a group, and their peers rated them, fill in the blank, whatever your peers rated you as. So it gives me information that helps me help you when you come ask me for a letter. Other questions? Anything? Content, exam format, questions, concerns?
I was very confused about this because I know you were explaining in lecture, and then you sent an email that you explained it wrong, and your email was a little confusing. When you were explaining about metaphase chromosomes and the spread, and they all look different, and they assume like different shapes, like some of them like X shape, and some of them are just like squiggly lines or whatever. So what is like the reason that they all look different? Is it proteins or?
So if I understand the question correctly, this is about the shapes of different chromosomes, all of which are at metaphase. So, which version of those two drawings are these chromosomes? Is this early in mitosis or late in mitosis? Yeah, this is early. This is while those double helices are glued together by cohesin. So each of those pairs of double helices looks like a thing. It's really two double helices lying side by side, and they're stuck together. Okay, the rest of the time when you see a metaphase spread, you see X's. That's later in the cell cycle. That's after the two double helices have been separated by separase and spread apart. I wish I could have recorded that and put that on the... So, all of what we just did will be uploaded like a normal video. And look on Blackboard later for information about the exam. So, we've seen on the first day or second day of class, you do a Google Images search for chromosomes. You see some karyograms. Each of these lines, of course, represents a double helix. You see some chromosomes that look like this, where the chromosomes are lying, the two copies of chromosome 1 from dad, let's say, are lying so close together along their entire length that they look kind of by eye like one object. And there are other times when those two double helices are drawn or look at metaphase like I've been drawing them so far today. They look like they're actually two objects crossing each other. They're X-shaped thing, which is actually two double helices. So if I interpret the question correctly, why is there a difference here? So this has to do with one of the study guide questions. The roles of the protein cohesin and an enzyme called separase. And this is one of the questions that one of you submitted for the study guide. So early in mitosis, cohesin glues, basically, the protein cohesin, glues the two copies of the chromosome together, essentially. So there's a point in the cell cycle at which a pair of chromosomes lying side by side look like a stick, is the best way I could put it. Then that enzyme, separase, comes along and cuts up all of the cohesin proteins. Then the arms, the two double helices are free to spread apart from each other. And that's when they start looking like that. So assume, for example, that the centromere is here. So early in the cell cycle, two double helices that are copies of the same chromosome, chromosome 1 from dad, let's say, get glued together by cohesin. Separase comes along and cuts them open, and then those chromosome arms can spread apart. Last question.
When you see the two sticks, is that the chromosomes in the stick form?
Right. So let's go back to the first. Start thinking of other questions to ask. Okay. So if I show you a metaphase chromosome spread that looks like this, let's start off easily. What do you think the ploidy of this organism is? Diploid, tetraploid. So this is during metaphase. This is after every chromosome has replicated. So if you're normally diploid, how many copies of each chromosome will there be at metaphase? 4. Okay, how many total double helices are shown up here? Right, each of these double-stranded chromosomes really looks like this. We just don't see this level of detail under a microscope. So how many pairs of chromosomes, how many groups of 4 are there? 2. There's a pair. They look like they're the same size and shape here. And here's a different pair. So this looks like a diploid individual. What would it look like during interphase? If this is metaphase, everything has replicated. There's twice as much DNA as there normally is during interphase. So how many double helices will there be in interphase? There will be 1 of each of these. There'll be 4. During interphase, if you're diploid, we've got 2 copies of each chromosome. So it'll be 4 double helices, 4 chromosomes during interphase. Just checking to make sure everybody's still okay. Now, what do you interpret it means if you see something that looks like this? So we use fish probes. What's the purpose of a fish probe? What sort of information does it tell us? Here's the review part.
The relationship between chromosomes.
Relationships between chromosomes. So does anything up here not make sense so far? This is your time to figure out where I screwed up. I did this on purpose, but what would you expect to see that's not here?
Maybe red on the bottom.
Right. If these two are homologous chromosomes, why haven't I drawn red dots here? Those fish probes give us information about how chromosomes are related to each other. So if you're diploid, you expect to see, well, Diploid during interphase. Again, this is looking during metaphase, so we have how many copies of each chromosome? Our 2 copies have replicated by metaphase, so we've got 4, 1, 2, 3, 4 double helices of each chromosome. So how many fish signals should we see? Each fish probe attaches itself to 1 chromosome, so we should see 4, like we do on the small chromosome. 1, 2, 3, 4. 1 dot, 1 fish probe, 1 chromosome. So why don't we see them on this chromosome?
It's either not related or it got deleted.
Great hypothesis, it got deleted. So there's a fragment of this chromosome that's missing. That's a reasonable answer. That's how cytogeneticists would interpret this experiment. Diploid, metaphase, expect to see 4 dots representing the 4 chromosomes that contain that specific DNA sequence. If you see fewer, you conclude it's a deletion. What happens if you see more? Could be a duplication. So what happens if you see something that looks like this in whatever, is that yellow? Help a colorblind guy out. Okay. So what happens if you see this? The yellow fish signals. Which pair of fish signals is the odd one out? Which one doesn't belong? Right? You've played that game before, right? This is the signals on the small chromosome. Right? Here, the fish signals are paired. The pair of signals is paired. They're on the same locations on the same arms of the homologous chromosomes. What does it mean when one of the 2, this one right here, has the signals and the other one doesn't? Suggests that maybe these duplicated. So maybe that section of chromosome I'm circling in red copied and pasted itself onto one of the small chromosomes. What's yet another hypothesis? Let's get more comfortable with having multiple competing alternative hypotheses that could all potentially be true.
Translocation.
So if there's a translocation, does that change the amount? This is a good review question. Does that change the amount of DNA in the cell? No, it just moves pieces of DNA around. So a translocation won't usually change the number of spots you see. So here we're seeing an extra pair.
What if the fish signal was like a really long yellow bar, but because you switched it over, so now it looks like 2 spots?
Ah. That's a reasonable question to ask as well. So let's see. What color can I use that I can see that I haven't used already? So what happens if you have, this is going to be difficult. But yes, you're right. I'm not going to draw it. But yes, you're right. If your fish probe is very long, then you could detect rearrangements that way. I won't put that on the exam.
When you're looking at something like this, do we assume that that's all the chromosomes, or can we give an answer like, what if it's not all the chromosomes that are seen? Does that make sense? So like,
Yes, that totally makes sense.
Okay.
Yeah, so the question is, if I draw something like this, let's assume we're talking about on an exam. Can you assume that this is all of the chromosomes that there are to consider? I'm perfectly happy to let you decide. If you want to make up some additional chromosomes and you draw them and you say, I think this happened, that's fine, because that shows me your understanding. If you do it properly. So if you draw some additional chromosomes and you say, I think this is the case, then I grade that question based on whether or not you explained it correctly, whatever it was you did. Right. I'm going to get in a lot of trouble because this is a big class and this is not a multiple choice test. And I'm telling you, which makes me uncomfortable, I'm telling you that I'm comfortable with you arguing your answers. And I will grade the questions that you answer like that. Sometimes there will be a correct answer, but a lot of times there won't, and if you come up with a reasonable hypothesis and it's justified appropriately, good for you. Sorry.
What is it, 8 double helices?
Pardon?
You know how you put 4 double helices? Isn't it 2 double helices per chromosome?
So what I was indicating here was that this is metaphase. This is not where ploidy is determined. So this is metaphase on the left. In an interphase cell where we're diploid, you'd have 2 copies of each of these chromosomes. So this double helix, that double helix, that double helix, and that double helix, for example, I'm saying move into this one daughter cell after cell division. So in an interphase of pre-synthesis, normal cell where we count, are you diploid, tetraploid, haploid, whatever, that's why we have 2 copies of every chromosome. But at metaphase, when we actually look at the chromosomes, when you do fish experiments, there's 4 copies, 4 double helices of each chromosome. Temporarily tetraploid. Temporarily tetraploid.
For the study guide one, the fish probe,
Yeah.
I have a question about, like, if you're undergoing fission, do you have to add a?
So, okay, so to make sure everybody's on the same page, so fission, so a chromosome breaking apart.
Right, do you have to add a centromere at the quote-unquote telomere then? Because doesn't, chromosome use centromere to replicate or whatever?
About
Yes.
this slide.
It seems like we're looking at half the process. Why do we look at half of it?
So the question is, which half of the process are you referring to? So you said we've talked about half of the process.
Kind of confused on that.
Okay. So we've talked about chromosomes replicating. So 2 double helices become 4.
Yeah.
Okay, that's synthesis, that's DNA synthesis. So as cells, we either have 2 copies of a chromosome, that's interphase, or we have 4 copies. That's after synthesis. So we have the cell cycle is you're either diploid, you've got 2 copies of every chromosome, or right before cell division, there's synthesis, we're temporarily tetraploid, we have 4 copies of every chromosome. This is always where we study as cytogeneticists, is we always study the metaphase, 4 copies of every chromosome part of the cell cycle. So is your question, why don't we ever look at the cell when it's only got 2 copies of the chromosome?
I was just wondering why it's 4 double helices instead of 8.
So the reason, so this is temporarily metaphase or temporarily tetraploid. So if any of you take anything away from this slide, it's that at metaphase, we have 4 copies of every chromosome. And during interphase, we have 2 copies of each chromosome. Cytogeneticists only ever look at metaphase. So anytime we look at a metaphase chromosome spread, it's going to be 4 copies if you're diploid. If you look in interphase, there will be 2. So the reason there are 8 double helices on this slide is because I've drawn 2 separate chromosomes. So let's call those chromosome A from mom. We have 2 copies of A from mom, 2 copies of chromosome A from dad, and 2 copies of chromosome B from mom and B from dad. So there are 8 double helices in this picture, but it's 4 of each chromosome, 4 of human chromosome 1 and 4 of human chromosome 2. So there are 2 distinct types of genetic molecules in this picture. 4 of the double helices are 1 chromosome. the other four double helices represent a totally different chromosome that's in this organism. We have to think about what's happening, which we haven't talked about in class. I was hoping some of you would bring this from earlier classes. We haven't even gotten to this chapter in the textbook yet, but this is critical. So, cell cycle. Growth 1, where the cell is making proteins, doing its normal day-to-day activities, taking out the garbage, making food, cooking dinner for the organelles, and so forth. Synthesis, where DNA is replicated. Growth 2, cell does more of its normal day-to-day activities. And then mitosis, or cell division. Okay. So one of the questions on that, I think the last question on the exercise was, where did these chromosomes come from in the first place? Most of you properly interpreted what I was very vaguely asking. So half of you about said, well, these chromosomes came from synthesis of previously existing chromosomes. That's correct. Where did all of my chromosomes originally come from? My parents. So the other half of you, or 2/3 of you, or 3/4 of you said, at fertilization, you get half of your chromosomes from your mom and half from your dad. Okay. So if you look in an interphase cell, this is kind of like at fertilization. When those sets of chromosomes come together, we're talking about interphase. Sperm meets egg, nuclei join together in this blissful harmony. Yeah. Okay, so at this point, you have chromosome 1 from dad, chromosome 1 contributed from mom. You've got, say, chromosome 2 from dad, chromosome 2 from mom. So the colors are different chromosomes, human chromosome 1 in blue, human chromosome 2 in red, one copy from each parent. Another one of the questions on that exercise was, how similar are the two red things to each other at the nucleotide or the DNA sequence level? Are they identical or are they not, in other words? Is the version of chromosome 1 you get from your dad exactly, to the letter, the same nucleotide sequence as the chromosome 1 you get from your mom? What do you think? There is a right answer to this one. It's no. So I can see a few heads shaking. The copy of chromosome 1 from you get, unless there's a lot of inbreeding going on in your family, copies of chromosome 1 you get from your dad and your mom are not going to be identical. They're very, 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 very similar. Last class I talked about the idea that when we started sequencing human genomes, we realized that there's about 1 nucleotide change for every 1,000 nucleotides in the genome. So if you compare your sequence and mine, we're 99.9% genetically identical. The same is true for our parents. Unless you're inbreeding, they're representatives of those random 2 humans that met and had us, the kids. So on average, our 2 chromosomes, the homologous chromosomes, are going to be reflecting that amount of divergence. They're about 1 in 1,000 base pairs different. And the same is true for the second pair, the red chromosomes, and so forth. So our chromosomes are very, 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 very similar. They're not identical. They're close. Okay. So here's the kicker. What ploidy are we at this stage? How many copies of each chromosome? How many copies of blue? How many copies of red? 2. We're diploid. So in interphase is where ploidy is defined. So you have to be looking in an interphase pre-synthesis nucleus. This is where we do actually have 2 copies of every chromosome, one that we got from our mom, one that we got from our dad. what does this same drawing look like after synthesis phase? How many red things am I going to draw after our 2 red chromosomes replicate? I should be more explicit. Each of these things, a chromosome is a double helix, which we've discussed. So let me make the drawing a little bit more explicit. What does it look like after synthesis? Every chromosome replicates, so now we have how many red double helices? 4. We get 2 copies of the red chromosome we got from our dad, and we've got 2 copies of the red chromosome we got from our mom. Each of them replicates. Why? Why is it necessary to do this step? What happens when we get to cell division, or mitosis? We're going to go from how many cells to how many cells after mitosis. You start with 1 cell, end up with 2 cells. What's the ploidy after synthesis has happened? There's a nucleus. How many copies of each chromosome do we have? 4. So this is where the confusion always comes. When we look at metaphase chromosomes, we've got 4 copies or 4n. We look, and we are, temporarily, tetraploid. We have 4 copies of every chromosome. So after synthesis, right, that 1 copy of dad's chromosome has replicated and become 2. So we started with 2 copies of red chromosome, now we've got 4. What happens after cell division is that the 2 resulting cells get 1 copy of dad's and 1 copy of mom's red chromosome. Chromosomes segregate so that each of the daughter cells looks exactly genetically identical to the cell that this whole process started from, a diploid cell. So we go in G1 interphase before synthesis, we're diploid. Synthesis makes us temporarily what? Tetraploid, 4 copies of every chromosome. After mitosis, we get 2 daughter cells that are once again diploid. So when is it that we look at chromosomes? I do not at all mind. This slide?
Oh, no.
If you have a chromosome, so I'm going to draw a single double helix, here's its centromere. One of the exam study guide questions was, what happens if this chromosome breaks at the centromere? So the centromere is pretty big, and you could actually break a, say, submetacentric chromosome in the middle of its centromere. So that each of the 2 resulting fragments each have some centromere left. So they go on and lead happy lives as chromosomes. They can be replicated, they can be segregated. Because they still have parts of centromeres, they can still be segregated into daughter cells appropriately during cell division. But what happens if the chromosome breaks here? And you get 1 double helix that's got the centromere, and you've got a little fragment down here that does not. What happens to this little bit of chromosome? That's the question, essentially, as I understand it.
Slops, isn't it? Because it can.
Right. So it has no centromere, so this part of the chromosome maybe gets replicated, but when cell division comes, there's no centromere for microtubules to grab and segregate the random fragments of DNA that are left. So those just get lost.
That would be the part of the question. The other part would be like, say, since it is a fission period, you broke it at the inverted, like say the P arm.
Yeah.
So I'm imagining that the centromere is only on one part of the chromosome, like say the Q arm, because you're going to invert the P arm. Does that make sense?
So if this inversion happens, what does this chromosome wind up looking like? So you just suggested invert the P arm. So I'm going that direction. So we draw the P arm upside down. There's some centromere here, there's some double helix. It attaches to the long arm. Am I heading in the right direction for your question?
It's not the way I answered it, so yeah.
What would happen to this chromosome? How many centromeres does it have?
2.
It's got one whole one, but it's got 2 physically separate half centromeres. So functionally, it has 2 centromeres, which is really bad for chromosomes. But that would be a reasonable answer to this question. This is what this looks like. If what you described happens, if it breaks in the centromere, the P arm inverts and then fuses back together. Other questions related to this? Follow-up questions, other topics? Go ahead.
Going